Before I forget, I would like to thank Cynthia Brink. If you could wave, Cynthia. For putting the whole conference together. Thank you very much. So we're going to move on to our first presentation that will be delivered by Hadil from York University. I won't even try your last name because I'll butcher it. The title of her presentation is Insect Biomass Trends in the Long Point Biosphere Reserve, Impacts of Anthropogenic Threats. Hadil is a PhD candidate in biology, working in Dr. Sheila Colla's lab at York University. Her current research is a collaborative project taking place within the Long Point region that focuses on the impacts of climate and landscape changes on insect communities within protected areas.
Good morning, everyone. Thank you for having me here to talk about my research. I did talk about this a couple of years ago at the Long Point conference when I was first starting out. And now that I'm further along and I've just wrapped up my last field season, there's a lot more to discuss. So insects are fairly important species or groups. They provide us with a lot of ecosystem services. And what ecosystem services really mean, it's basically any service we as humans get from the environment that benefits us and that we rely on. So one of the more important services that we rely on is pollination. Insects pollinate almost 1,500 agricultural crops, and without this pollination service, we would have, we would have an issue with food scarcity and food security. Some other services is pest control through integrated pest management, as well as nutrient cycling, where certain insects break down detritus and release nitrogen and phosphorus back into the system for reuptake by plants. Because of their host plant specializations, butterflies and moths are great indicators of environmental quality. For dragonflies, mayflies, stoneflies, and caddisflies, all of these rely on water, specifically moving water during their nymph stage for habitat, and because of this, they're excellent wetland health indicators. So we've talked a bit about how insects are really important. I'm going to get into insects and why and how they're declining. So on the left, the study by Hallman et al. In 2017 focused on 27 years of insect research in protected areas and found that about 76.7% of insects have declined within these areas within that 27 period. Another study by Siebold et al. In Germany, also this time focused on grasslands and forests, found that both biomass of insects and species diversity decreased over just a 10-year period. So even though we rely heavily on insects and there's a lot of decline that's been proven, not a lot of funding is given to insect research. So on the pie chart, you can see that arthropods make up the largest proportion of species richness in the world. But if you look at the graph on the right hand, you can see that per species, arthropods, mainly insects, receive a lot less funding than other taxa, primarily vertebrates, because they're megafauna, there's a lot more focus on them, they get a lot more money, and that's just like the sad truth of it. So, as we all know, protected areas are very important areas. They're habitats that, they provide habitat for endangered species and are basically unfragmented natural ecosystems. So this is just a map of all the terrestrial and marine protected areas in the world. And these areas only make up about 17% terrestrial and 8% marine. Even though there's such a small area, there's so much dependence on these areas to protect all of our at-risk species and all of our biodiversity. So I'm gonna get straight into talking about my research. I'll get into the history of it a bit more, but before I talk about my sites, back in the 90s, Peter Carson set up a bunch of traps at all of these sites in the Long Point area, primarily the National Wildlife Area, and he collected insects from these sites, and they've just been sitting in storage for 30 years, so that's really the basis of my research. And on top of that, Michael Bradstreet did manage to collect a lot of vegetation data from 91 to 2021, so over a 30-year period. So I have about 11 sites. Most of them are located within the National Wildlife Area. The different colors just denote what age the dune is, and I'll get into more of how the dunes look in a bit. So we have older dunes, younger dunes, intermediate, and one site labeled as other. So I have about 6 older sites. These older sites are basically all savannas and woodlands, and they're mainly dominated by red oaks. We've got 3 younger sites. These are mainly lightly vegetated sand dunes with cottonwood in them. There's one intermediate site, so this is a coniferous woodland, and it has interspersed wetlands throughout. And then our last site, which is labeled as other, and this is mainly because an age wasn't assigned to it because it was disturbed by a fire in the 60s. So the main research question I have is, how have insect communities within protected areas changed between the 2 time periods, back in the 90s and currently, and what environmental variables are really driving these changes? So more to give you a better idea of what happened in the 90s, like I said, Peter Carson set up a bunch of malaise traps at all of these sites in the 90s, primarily 92, 94, and 95, which are the sites that I'll be focusing on for the historical period. And he set them up using, I don't know if you can see them in the picture, but they're permanent T-bars that he staked at each of the site and just hooked up the malaise traps to them. And like I said, Michael Bradstreet and a few others have been collecting vegetation data at all of these sites within that time period. I should have mentioned this before, but each site is within a different breeding bird census plot at Long Point. So for my methods for comparing how insect biodiversity and the community has changed between now and then, I've basically set up malaise traps at the exact same location, facing the same direction that Peter had set them up in back in the 90s, and that was easy. It was just mainly going out and finding where the T-bars are and like hooking up the malaise traps to them. And we sampled each site for the exact same number of days as the historical site. And instead of copying the calendar date that they were started sampling historically, we calculated the number of growing degree days and tried to match that up with the historical sampling. So these are just jars of what a sample from a malaise trap would look like. And our methods were basically sorted through this jar down to insect order. We weighed each order as a wet sample, and then we dried each order and weighed it again as a dry sample. So for the historical samples, because they've been sitting in storage essentially for 30 years, it's a prolonged period of time, and research has shown that prolonged storage of insects, really arthropods in general, over time in ethanol showed a decrease in biomass because the lipids, the fats in these organisms tend to break down over time. And a few papers were able to test how much content of lipid made up insects, and they came up with 11.8% approximately. So because of this, we adjusted our dry weights of the historical samples to account for this fat breakdown, so things would be more comparable. So for the variables that so far I've tested, and so this is all just preliminary results, things will likely change as time goes on. We started out with 20 vegetation variables on different types of vegetation, mainly richness, species count, abundance, and things like tree diameter as well, and stem counts. So our models couldn't handle 20 variables of vegetation. So we ran a PCA, and because so many of the variables were collinear, we narrowed down that number from 20 to 9 variables. And for climate variables, we mainly used these 6, and we used them consistently throughout. So frost days and winter precipitation basically are the values from the winter previously. So if we had sampled for 1992, we would be looking at the winter from the previous year for the number of frost days, how many days the temperature was below 0, and winter precipitation is the amount of snow that was deposited. And then summer precipitation, how much rain we got, and then the mean max and mean minimum temperature are the means for the summer months, and then the growing degree days that I mentioned before. So all of these are preliminary results. There wasn't much variation between sites, so all of the data that I'm showing here are combined totals of dry biomass for all 11 sites, and I just grouped them by the different time periods. So we can see that, before I forget, most of the data that I'm showing does not include Hymenoptera, just because in the historical samples, a lot of the bees were removed and never returned. So to make things more consistent, I'm not including Hymenoptera in any of the data. So we can see that from the 90s to today, there's about a 200-gram decline in dry biomass. And this decline was mainly driven by the number of frost days. It was the only significant variable. So to see if maybe this decline was more apparent in aquatic versus terrestrial groups. So aquatic insects are any taxa that have an aquatic life stage, and then I just grouped all of those together, and then the terrestrial ones are all the groups that only had terrestrial life stages. So you can see that there was a decline in both groups. It was a more apparent decline in the terrestrial groups. The mean minimum temperature for the mean minimum temperature was the one that was most significant for the aquatic group, and the number of frost days was most significant for terrestrial, but they both showed a significant decline in dry biomass. So when we look at the individual orders, only 5 of the orders were significant. 3 of them were terrestrial, so the Lepidoptera, Coleoptera, and Brachycera. And then Nematocera and Neuroptera were the significant aquatic groups. So each of these was only significant with various climatic variables. None of the vegetation variables were significant for any of the models that I ran so far. It is worth noting that Brachycera and Nematocera are suborders of Diptera, the flies. It's just Brachycera are more terrestrial and Nematocera are more aquatic, so that's why they've been separated out. So just a quick recap. So far for our preliminary results, it seems that dry biomass of insects has declined between the 2 time periods, about a 200-gram decline. 200 grams might not seem like a lot in theory, but if you think of it in terms of midges, so 1 midge is about 0.5 micrograms, so that's essentially a 400,000 decline of midges between the 90s and today. And most of the decline that I've seen so far has been driven by various climate variables depending on the taxa. I'm still running more tests, and I have to run a few more models, and likely compare the sites in a different manner to see if anything pops up there. But it's still ongoing, it's still preliminary. I'll likely have more results in the next few months. So yeah, my next steps are to identify specific taxa down to species level and try to compare diversity and see if there is any changes in that between the time periods. And like I said, more site level comparisons. So I just really want to thank all of you for listening today. York, NSERC, and the Entomological Society of Canada for funding this. Michael Bradstreet and Peter Carson for all their help with the historical data, answering all my questions. And obviously, Canadian Wildlife Services and the Long Point Bird Observatory for help with planning all of this and site access. Thank you again for listening. I'd be happy to answer any questions.
Excellent. Thank you. Do we have any questions?
I just wondered about wind effects, whether the Malaise traps might have been more protected in one time period or the other.
I haven't looked at wind yet, but that's the next variable that I wanted to include. I think it's likely different just because of the way the vegetation and the landscape has changed between the two time periods.
Another question?
Thank you, great presentation. You mentioned that one of your plots was burned, so I was wondering if you could talk a little bit more about the differences between, burned and unburned plots or traps.
I can't really say much. When I looked at the differences in the biomass across all sites, they all showed a decline. Nothing was more significant than the other sites. It was all pretty much the same, so maybe once I try and group them based off of age rather than individually, things might pop up as different.
Thank you.
Question back here?
Can you hear me from here?
Yeah.
Yeah, okay. So I'm just wondering about what you saw with the dry biomass decline with frost. Was that in response to less frost days?
Yes. So in the 90s, the average number of frost days for that group of years was about 86. And the average number of frost days for when I sampled, which was 2021 to 2023, was about 74 frost days.
So I'm just wondering why you think that relationship is. Like, why would less frost days lead to the decline?
It might be an earlier emergence of insects, so that's why it's not showing up. It might be popping up earlier than when we sampled, but that was the point of the growing degree days, to make sure we had the same number of accumulated days when we sampled with when they sampled, but it was still a decline, so it's likely other climatic variables that are also pushing this, but only frost days that was the only variable that popped up. You're welcome.
Yes, sir.
Yes, the German paper, I think they attributed to a particularly harsh use of pesticides, insecticides, and you were pointing at neonics. I didn't see you make mention of anything to do with possible effects of pesticides and drastic use of white.
So
Could you repeat the question, please?
Yeah, so the German paper, the Hartmann et al., one of the variables that they looked at was the use of neonics and pesticides, and I didn't really focus on that in mine. So mainly because all of my sites are within large protected areas, so here these sites were all within the National Wildlife Area. They haven't been using, there is no pest use or neonics use there. They have been spraying for frag, and I'm hoping to include some of that data later on as I do more analyses.
Any further questions? One at the back. Can you come up to the front, please?
Sure, I'm also really loud.
We'll move this mic about halfway back here during break.
Super cool work and very important. I had a question. Is anyone, or including yourself, planning on looking at the DNA, or is it like super hard to extract?
So that was one of the main things that we wanted to do. We wanted to do eDNA on the ethanol rather than like pulverize all these insects, especially because there's like thousands and thousands of insects. I've had 150 jars of insects just to like work through. But the way the historical samples have been stored and the way my samples have been stored isn't very conductive for DNA samples. So once you remove the sample, you have to freeze it right away and store it in the dark just so the DNA doesn't get denatured. And that didn't happen. We were too far along before we thought of that, so we can't really do DNA sampling.
Thank you.
Thank you.
Our next presentation will be by Natalia, who is a second-year master's student in the Department of Geography and Environment at Western University. She is conducting her work with Dr. Jed Long and is interested in spatial ecology and wildlife management. Today, she will be presenting her findings to date on her master's research. And the title, of course, is Understanding the Spatial Ecology of Coyotes within the Long Point National Wildlife Area.
That's great. Hello, everyone. Hi, I'm Natalia. I am a second-year master's student at the University of Western Ontario, and I'm doing my work with Dr. Jed Long in the Department of Geography and the Environment. And today I'll be speaking about kind of what I've found so far. Specifically, I'm looking at the spatial ecology of coyotes within the Long Point National Wildlife Area. So coyotes are one of the main apex predators within the LPNWA, and they can, as a result, exert negative top-down pressures on prey species, and in turn allow for the management of prey populations. And this is something that the Canadian Wildlife Services is particularly interested in because historically there's been an overabundance of white-tailed deer within the site, and there has been extensive management of this population. And they're interested in understanding if coyotes are able to sort of take over the management of this population and act as a primary predator of white-tailed deer. So my work is hopefully going to inform baseline information about coyotes and what their role is within the LPNWA as a whole. So within my thesis, I have three main objectives. The first is to understand the spatial temporal dynamics of coyotes within the LPNWA. The second is a bit more informal, and it's to help inform a coyote trapping plan that we'll be going through or has already gone through. And then finally, quantifying the abundance of coyotes within the LPNWA. So within this slide deck, I won't be talking about abundance, but I will be going over resource selection of coyotes within the LPNWA and our coyote trapping so far. So in order to study coyotes within the LPNWA, I need to be able to collect data, and the choice of collecting data that we're using is camera traps. So camera traps are remotely triggered cameras that take a photo whenever an animal walks in front of the camera. And it's a really great way of collecting data because it's relatively non-intrusive, and when it comes to collecting data on a highly elusive and low-density species like coyotes, camera traps are great because you can actually get data on them. They're not like turtles where you can go out into the field and catch them, so getting photos of them is one of the best things that you can do realistically. And the way that we have our data collection set up is that we have 90 possible sites within the LPNWA, and they are located proportional to the different habitats available and within a 500-meter hexagonal grid. And this just allows for us to both sample all the habitats within the LPNWA as well as get a good spatial resolution of the LPNWA as a whole. And although we have these 90 sites, we only have 30 cameras, so we rotate our cameras approximately every 3 to 6 months. So since June of 2022, when we started this project, we've collected over 600,000 photos, and this is from 78 of our 90 sites, We just recently collected the data from the last 12, but I haven't had the time to go through it yet, so it will be eventually added in. And this amounts to over 12,000 camera trap days. And 600,000 sounds like a lot of photos, but realistically, the vast majority of them are empty. So within most camera trap studies, about 70% of photos are blanks. So that means that they were photos that were triggered by moving vegetation, and there's no animal in the photo. So a big part of the first years of my master's was trying to figure out how to go through my data efficiently so that I could get the data out of it and finish my master's in a timely manner. And what I ended up finding was the best was utilizing Megadetector. So Megadetector is a Microsoft-based open source AI that will allow for you to feed in your images, and then it produces a confidence interval between 0 and 100 of whether or not it thinks that there's an animal in it. So this video of images, it's not from Long Point. No tigers yet. But it kind of shows how Megadetector works. So it creates a boundary box, and it says where it thinks that there's an animal. And utilizing this, I've been able to reduce that number of 600,000 photos to about 200,000. So about a third or 2/3 were eliminated, which is great news for me. Now with those 200,000, they don't necessarily all have animals in them. There is a bit of error within that. So I have to go through the photos again, and about, I would say 70% are still blank. But any of the ones that do have animals, I will then go and assign a species, an age class, and then the amount of individuals within each one. And now we can get into the data that's actually been collected. So far, the top 3 species that we see on our camera traps within the LPNWA are coyotes, white-tailed deer, and wild turkey. So we have approximately 3,000 photos of coyotes, 44,000 photos of white-tailed deer, and 5,000 photos of wild turkeys. And something to note is that although the number of 44,000 deer compared to 3,000 coyotes seems pretty large, deer have a habit of going in front of the cameras and finding a bush and then just sitting there and eating that bush for a half hour. So there's not actually 44,000 photos of different deer, whereas coyotes, they kind of just run past. So with that data, we then go through it and we make sure that all of our observations count as one group or one individual going in front of the data. So generally, we'll put a 60-minute buffer, because that's around when we'll see a new individual come across. So using this data, we can then start to look at what the coyotes are actually doing within the LPNWA. So here I have a map of the relative abundance index of coyotes from our cameras. The relative abundance index is an index that's often used in camera trap studies, and it essentially means the amount of observations at a site divided by the effort put into that site. So in this case, the amount of coyotes in one of the camera trap sites divided by the amount of days that the camera was up. And utilizing these kinds of maps, we can start to kind of predict what we think is affecting where we see more coyotes in our images. So just looking at this map overall, we can see that there seems to be these larger squares around the shoreline. From being on the LPNWA, you can see that they tend to use that shoreline a lot, almost like a highway to get around. Another thing that you can tell is that almost all of the dots are filled in, so meaning that we have photos of coyotes basically everywhere. So coyotes are really actively using the entire site. We can also then try to kind of pick apart more habitat-based features, so they seem to be in areas that look like they have less vegetation, so more so in the dune systems rather than in the forest areas. And this leads into the first part of my master's work, which is trying to understand the resource selection of coyotes. So what's actually, using habitat variables that I can collect from imagery, what of these variables then affects how many coyotes we're seeing? So we're trying to understand why coyotes are in specific places compared to others. And some of the variables that I'm interested in include seasonality, the vegetative cover, the terrain roughness. So are they more likely to be in areas where it's super hilly, or do they not want to climb all those hills? Edge effects. This is something that a lot of coyote papers look into because they like to use edges, so edges of forests, to forage for animals. Distances to the lake or distances to wetlands where there might be more prey, as well as things like mean habitat area. So if an area has a lot of forested area, is that where they're going to stick close to, or perhaps the beach? So in this, I'll be talking about specifically 2 of my findings so far. So I'll talk about the vegetative cover and seasons and their effect on coyote presence. So NDVI is a metric often used to understand vegetative cover. Essentially, what it means is it's the normalized difference vegetation index, but in simpler terms, it's kind of more a measure of greenness. So the higher the value, the more green the area is. So you can imagine like a dune system will have a lower value versus a forest. And what we found is that generally coyotes seem to be preferring areas with a lower NDVI, so less green areas. Or at least this is where we're getting a lot of photos of them. And we see a decrease in their relative abundance as we get to greener areas. And then when we look at seasonality, we can see that the RAI, the relative abundance index, is higher in colder months, so in the winter and the fall, than it is in the spring and the summer. And this makes sense when you're thinking about coyotes, because in the winter and the fall, that's when they're dispersing and they're breeding, so they have to be moving around a lot more. As well, in the winter and the fall, there's a lot less resources. So the amount of traveling that they have to do to find prey items is quite a bit larger than, for example, in the spring and summer, when they're pup rearing and there's quite a few more resources around. They don't necessarily need to or want to be moving around as much. So with this part of my thesis, I will be adding those additional variables that I'm interested in into understanding what affects where we're seeing coyotes, and then hopefully putting in a bit more effort into understanding what might be accounting for these differences outside of habitat features. So for example, the camera detection, generally you can see for longer if you're in a more open area, like a shoreline, than you are when you're in a forest. So just really fine-tuning those details and being able to understand the resource selection. And now I'll move into the coyote collaring. So this was a bit of an informal goal of my project. Back in the 80s, this was when the last time the coyotes were researched, and there was some effort put into collaring the coyotes. However, they were unsuccessful. So with the interest in understanding coyote ecology within the LPNWA, there was a want to collar. However, they didn't want to put in the effort to collar the individuals if it wasn't going to be successful. So using the relative abundance index and then looking at the photos and trying to understand the general pack sizes and where we were seeing movement, we developed a trapping protocol to try to trap some of these individuals. And we set up to do this in September and October, and we were successful, as the images show. So we had captured 11 individuals, 7 of which were outfitted with GPS collars. In the top photo, you can see one of the individuals that we've caught. We put them under while we're processing them to ensure less stress. And you can see Dr. Judd Long, Samantha Lalonde from the CWS, and Tyler Wielden up there with me. Tyler is a research biologist with the MNR who has tons of experience doing this. And it was really cool to be a part of it, but something that was even cooler was that we did this in September and we went back out in October, and within 3 weeks' time, we had gotten videos and photos of our collared individuals, which you can see here. So this data is exciting because it will help to inform the next step of this project, which will be more so understanding the interactions between coyotes and deer within the LPNWA. And the GPS data will be pretty influential in this because then we can see specifically where these coyotes are going and areas that they're hanging out in. When we came back in October using this GPS data, we were able to visit some of these areas where we saw that the coyotes were hanging out, and we found some evidence that there were deer carcasses there at some point, which is exciting and is perhaps evidence that they are actively predating the individuals, but all these details will have to be, fully explored by a PhD student. So if anyone knows anyone who's interested, Dr. Jed Long is currently looking for people. Yeah, and I just want to say a thank you to anyone who's helped with the camera trapping. And not pictured here is Danny Bernard, who has been pretty essential in the logistics and getting the project going. And then I have included some images and videos that I thought were particularly interesting, but didn't really fit in the presentation. But that's all. Thank you very much.
Thank you, Natalia. Questions? Come up to the mic, please. Thank you.
Great presentation. I was just wondering in regards to CWS hoping for the coyotes to perhaps take over the deer management. Is there any aspect that you're going to look into as far as like whether they look like they hunt in packs at all? Because I know like some areas coyotes do that, and some areas they don't, and when they do, they often hunt more deer.
Yeah, so that's going to be, I think, a question that the future PhD student will be focusing on. Sadly, with my timing, I won't necessarily get to work with any of that GPS data. I'm primarily working with the camera trap data, but I think it will be really interesting to see if the coyotes do hunt like packs as they do more northeast of here, or if they more so take advantage of fawn season, and that's when they're able to reduce recruitment of individuals.
Any other questions? Yeah, both of you come up from the back. That'd be great. And anybody else has a question, we can probably squeeze 3 in here. Thank you.
Super cool work. I love seeing the videos. I had a question, and this might actually already be part of your next steps. But in terms of, have you just done a simple analysis where you're looking at your sort of false positives, like when the algorithm says that there isn't a detection? Have you just looked at that rate and then split it out by your different variables, like the amount of vegetation, just to see if you like see, for example, there's more false positives if there's a lot of like greenness, for example?
When you say false positives, do you mean in the site itself?
Yeah, so like when it says that something's being detected and then you actually go to look and there's nothing there.
For the images themselves?
Yeah.
Yes, I have. So a big part of my first year was that I did an analysis with about 30,000 photos from 13 different sites. And I ended up going through and tagging all the photos by hand, and then I put it through Megadetector. Yep. And I looked at the comparison of if it missed any. And generally, as long as I kept my confidence level very low, which I currently have it at 0.1 or 0.01 out of 1, then it didn't miss any. So it's fairly rigorous at being able to detect when there's an animal in there.
Okay, cool. Thanks.
Thank you.
Well, just one quick question. I was just wondering what would be the most unexpected animal you would have seen on your photos?
There have been some photos of rabbits, which when reading one of the papers from the 80s, they were saying that there weren't any rabbits on the LPNWA, so that was cool to see. And then there's possibly a photo of a fisher, but
Madeline.
no badgers. Lots of opossums and raccoons.
Hi, my university. Are there any students comparing this kind of work on the other parks? I know years ago deer were overrunning Peely, and conservation officers with gun licenses, I suppose, had to cull every so often to keep the balance. Do you have any information on that?
With that specifically in parks in Ontario, I'm not too sure. I think that generally there is an issue with overabundance in deer because their main predator that used to exist here, wolves, don't exist anymore. But there are quite a few papers that look at smaller management areas, specifically in the US, that found that coyotes were able to reduce the populations of deer, primarily through hunting them in the winter, as well as going after the fawns during fawn season.
And any further questions? One comment. Please archive your data really, really well. Can you imagine you do this study in the next 20 years? Excellent. Thank you.
Thank you.
Well, it is now time for break. And just behind these walls here, we have some excellent posters. So I encourage you to wander around, and we have displays behind the wall. And we shall meet back here at about 11:30. Thank you.